Today I will be explaining the synapse of chromosomes in meiosis using two individuals, which is labeled here. Looking at this chromosomal aberration, it can be determined that this is a pericentric inversion because the movement of genes was changed across the centromere. Additionally, we have to understand that our chromosomes will have to compensate for the inversion that takes place, which will be represented by a loop that compensates the inverted region located in chromosome 2. Lastly, we will have to rearrange the gene order in chromosome 2 to represent the homologs lining up together for an individual outside the brackets. Starting off with our first chromosome, we have an extended omega that bubbles out to accommodate our future loop. Keep in mind that we have two sister chromatids for meiosis, which is labeled in the drawing. Now we can draw a chromosome 2 as our loop labeled in the drawing. Like chromosome 1, chromosome 2 also has a sister chromatid, which encases our loop. We can go ahead and add our genes to our two chromosomes. We will start to the left where M is labeled to the outside of the loop. Within the inversion, our genes are labeled as I, J, followed by our two centromeres K and L. Lastly, H and G are labeled to the right side of our loop. Now that we have our genes labeled, we need to understand that a single crossover occurs between the J and K loci, which is drawn out in our loop. I will now go ahead and illustrate each gametic pathway of our pericentric loop with a different color and a fun example of a dog running through our drawing for better understanding of the material. For our first gametic pathway, our dog example in the color red represents the pickup of M, I, J, followed by centromere K, L, H, and G. For our second gametic pathway, our dog in the color blue represents the initial pickup of M, I, J, and a centromere. We now face our first crossover between J and K, where our dog will cross over to the loop and run back to the left side and pick up K, L, and M. For our third gametic pathway, our dog in the color green represents the pickup of M, running up to L, K, or centromere, J, curling down to I, H, and G. For our fourth and last gametic pathway, we have to start in the upper right hand side because we already have used our other three pathways for our inversion example. This pathway will start off with our dog in the color orange representing the initial pickup of G, H, L, and K. We now face our second crossover between J and K, where our dog will again cross over to the loop, but run back to the right, picking up a centromere, J, I, H, and G. Here are our four gametic pathways with the respective color from each pathway. We can see from all four pathways that gamete 2 and 4 are not viable gametes because they do not contain all of the respective loci and either have a duplicated locus or duplicated loci. For individual 2, I have listed the following chromosomes. As we can see, these chromosomes are mirror images of one another, so we can match them up by flipping one of the chromosomes. Since there are no aberrations in either one of these chromosomes, we can infer that each of our synapses will result in chromatids that follow a linear path, even with two crossover paths between J and K. Listing our example for individual 2, we can draw each pathway with the colors red, blue, green, and orange. The pathways of the colors blue and green represent the two crossover points between J and K. The resulting chromatids from each pathway are all viable because they contain all respective loci in the centromere. Between individual 1 and 2, we consider individual 2 to be more fit because it produces our 4 viable gametes in individual 1, who only produces 2 viable gametes. Thank you for watching this video, and hope this has helped with your understanding of chromosomal aberrations during meiosis.